Hello viewers, welcome back again to yet another video on special senses, particularly the eye. So in this video we're just going to continue from where we stopped. We stopped on photoreceptors while we're doing the cones and rods. We talked about their structure and everything. Now in this video we're going to talk about phototransduction there. So basically what we're just trying to do here, we're just trying to see how light energy is converted to action potentials that are then sent into the visual pathway and therefore sent to the brain for perception of vision to occur. So what is phototransduction? So in just simpler terms, phototransduction, I can say light energy to receptor potential in photoreceptors. So what do I mean by that? Light energy to receptor potential in photoreceptors. All that I'm just trying to say is that this is the process by which light energy is converted into a receptor potential in photoreceptors, which are the rods as well as the cones. Now, what is a receptor potential? What is a receptor potential? A receptor potential is a non-propagated, take note of that, non-propagated transmembrane receptor transmembrane potential difference, I would say voltage across a membrane. Now, what do we mean by non-propagated transmembrane potential? If by this word non-propagated, we mean that this receptor potential does not travel from one point of a receptor to another. For example, if this is generated in this, if this receptor potential is generated in this receptor here, it won't be able to propagate from this receptor into another cell. It doesn't move. Then transmembrane potential difference, it just simply means the voltage across the membrane due to the difference in the inside and outside of the cell due to the electronegativity difference that is brought about by the difference in anions as well as cations inside and outside the cell. That's what we have there as a receptor potential. What we should take note here is that this receptor potential is not an action potential. It is not an action potential. In other words, what we're just trying to say that in other cells, you end up having an action potential, that is a disturbance across the membrane, or would say a series of events that will take place due to the movement of ions and anions, across the membrane. But now this receptor potential is not an action potential, it's just a graded potential. What do we mean by graded potential? I would say GP is a graded potential. It means it's a local, meaning if it's starting at this point, it will remain at that particular point. It's local and it's mild. It's just a slight change in the membrane potential. So what we should take note is that in most sensory receptors, when those sensory receptors are stimulated, they get depolarized, and this depolarization is going to lead into an action potential. That action potential is going to lead into the release of neurotransmitters at the synaptic terminal. However, in these photoreceptors, we are not using action potential to release neurotransmitters at the synaptic terminal. What we are using are the receptor potential, which are graded potential, the non-propagating locally mild membrane potentials are the ones that we're going to use to release neurotransmitters. Now, let's talk more about these photoreceptors. If you remember from our previous video on photoreceptors, we talked about cones and rods, and in this video we're going to use cones, but you should remember that the same concept can also be applied when you're talking about phototransduction in cones. The only difference that we're going to put is just the photosensitive compound that we're going to be using. Now, last time we talked about these having the outer segment and as well as the inner segment. Now, this part here on top is our outer segment. And for the rods, we say that these outer segments have got membranous discs. In these membranous discs, these rods have got about 40% rhodopsin or visual purple. So this rhodopsin is a photosensitive compound that gets to react to the presence of light, and it's the one that helps in phototransduction. So it's also known as visual purple, and it is a combination of a protein component, called scotopsin, as well as vitamin A or retinol. Now, this retinol is in the form of 11-cis retinol. It is actually gotten from, it's not synthesized in the body, it's gotten from those food products that have got beta carotene in them, such as carrots. So it is in the form of 11-cis retinol because this is the only form that has the ability to bind with scotopsin. Therefore, they end up forming rhodopsin. When this 11-cis retinol binds with scotopsin, it gets to form rhodopsin. So we should remember that this one is found here in the membranous discs of the rods, and is found in the circules. In the circules of the cones. You have other photosensitive compounds which are not rhodopsin, which are the ones that we talked about in the previous. So the way we are going to talk about this phototransduction, we're going to do it in two ways. We'll first talk about how receptors are in the dark when the person is in the dark, and what happens when there's actually light that has been shown into the eye of an individual. So here what you are seeing are just, yes, it might look confusing, but they're just simple diagrams, which I will explain as we move on in the video. Here it's just showing how these photoreceptors are arranged in the retina. If you remember, we talked about the layers of the retina. So this is actually a rod, which is synapses with the bipolar cell here. This is a bipolar cell, alright, BP, and this is a ganglionic cell. So the bipolar cell synapses with the ganglionic cell. It is in these ganglionic cells that an action potential is generated. We should take note of that. Action potentials in phototransduction are generated in the ganglionic cells of the visual pathway. Then in bipolar cells and rods, you only have receptor potentials. Non-propagating voltages across the membrane. Now, this is another part. If you zoom in here on the outer segment, you're going to have this. So this is our outer segment, and this one is the inside of the cell. Sorry, this one is the outside. This one is the inside. This one is the cell membrane. Remember, we said in rods, in the outer segment we have discs. So this one is a disc, membranous disc. So this is the membranous disc, the one we're talking about. We said in the rods we have membranous discs, and I said in those membranous discs that's where you are finding photosensitive compounds. And this here on top is a photosensitive compound rhodopsin, which is in the rods. So this is rhodopsin. Rhodopsin. This rhodopsin is actually coupled to a G protein. It's actually coupled to a G protein, which is known as transducin. It's a G protein, transducin. Then this G protein has got three. If you can see the way the rhodopsin is like this, then there's 1, 2, 3. It has got 3 subunits: the alpha subunit, the beta subunit, and the gamma. So this one is the gamma subunit of the G protein transducin. This one is the beta subunit of the G protein transducin, and this one is the alpha subunit. The other thing that you're going to find in the membrane of this membranous disc is this enzyme here in green, known as PDE. PDE means phosphodiesterase. Phosphodiesterase. This is the phosphodiesterase enzyme, PDE there. Then the other thing that you're going to find that is membrane bound is GC. GC there for guanylate cyclase. Guanylate cyclase. It's also a membrane bound that you're going to find bound to the membrane of this membranous disc in rods. Now remember, we're talking about this during the dark when there's no light. So what we're going to do here, I'll just create space by removing this stuff here. I should take note that GC there is guanylate cyclase, PDE is phosphodiesterase. Then take note that here is rhodopsin, the alpha subunit, beta subunit, and the gamma subunit of the transducin protein, which is a G protein. It's a G protein there. So during the dark, these photoreceptors are depolarized. Photoreceptors are depolarized. Now remember, I said that when sensory receptors have been depolarized like this, they get to release neurotransmitters. So what we can say is that during the dark, as since this photoreceptor rod is depolarized, it gets to release neurotransmitters on the bipolar cells. So those are neurotransmitters, and this neurotransmitter that they release is known as glutamate. Let's hold on to that thought for a while. We'll come back to it. But what we're saying is that these photoreceptors are depolarized and they are releasing glutamate between the synapse between rods as well as the bipolar cells are releasing glutamate there. We'll take hold onto that. Now, how does this depolarization happen during the dark? Before we explain that, the first thing that I'm going to talk about is role of guanylate cyclase. Now, remember we said we have got GC, guanylate cyclase, which is this enzyme, which is membrane bound in the membrane of the membranous discs here. So guanylate cyclase, what it does is that it converts GTP, guanosine triphosphate, into CGMP. So it converts GTP to CGMP, which is cyclic guanosine monophosphate. Converts GTP, so here you have GC, which is guanylate cyclase, converts GTP into cyclic guanosine monophosphate. That's what happens in the dark. So in the dark, what we are saying is that the levels or the concentration of cyclic guanosine monophosphate goes up in the dark. It gets to increase in the dark. So due to that, this binds on this channel here. This channel here is a cyclic guanosine monophosphate gated cation gated channel. So in other words, the binding of this cyclic guanosine monophosphate on this channel keeps the channel open to allow cations to influx, to allow cations to enter into the cell. So since we have explained this, we'll go back to this, we'll go to this diagram. So now let's take a look what happens. Cyclic GMP has increased in the inside of the inside of the cell on the outer segment of this rod photoreceptor, and this is due to the action of guanylate cyclase, which converts GTP into cyclic guanosine monophosphate. Cyclic guanosine monophosphate binds onto this channel here, cyclic guanosine cation gated channel, and the binding of that keeps that channel open. Now, remember I said this photoreceptor is depolarized. In most sensory cells, most sensory receptors, the resting membrane potential is usually from in the range negative 70 millivolts to negative 90 millivolts. However, that is not the case for photoreceptors. These photoreceptors, in this case we're talking about the rod, is during the dark state, it's in a depolarized state, and the membrane potential is about negative 40 millivolts. This is constant, and it is called the dark current. It's called the dark current. Now let's see how this comes about. And for us to talk about that, we're going to use this diagram, and let's see what exactly happens. And I'll start from the fact that I'll start from this point here. I will label it as A. What is A there? A is actually the sodium potassium pump, or the sodium potassium ATPase. This one in the photoreceptor just maintains the concentration of potassium and sodium inside the cell. Just functions to maintain the concentration of sodium inside and outside the cell. So what it does, that it moves out 3 sodium ions into the outside of the cell, as you can see by that arrow, and moves into potassium inside the cell there. Now, there is also another channel here at B, which is a non-gated, meaning it's not closed, non-gated potassium channel. In these two, the sodium potassium ATPase and the non-gated potassium channel, are in the inner segment. So going down here, inner segment, going up there, outer segment. So these two are in the inner segment, and this non-gated potassium channel, what it does is that it takes out potassium ions from the inside of the cell to the outside. It leaks out potassium channels like that. Remember, we've already leaked out sodium ions, and we've taken in potassium ions using the sodium ATPase, so the sodium potassium ATPase pump. Now this one, the non-gated potassium channel leaks out potassium potassium from the inside to the outside here. Now, this leaking out of potassium, the efflux of potassium from the inside to the outside opposes the leaking or the going back of the sodium into the cell through these channels here. Remember, we said these are the cyclic guanosine monophosphate binding here, and these channels are open. Those channels allow sodium to move into the cell like that, as well as allowing calcium ions to move inside the cell. So the movement of these sodium ions and calcium ions into the cell depolarizes this cell, while the moving out of potassium acts to oppose this depolarization by hyperpolarizing the cell. So in other words, this depolarized state of negative 40 millivolts of these photoreceptors, would say it's a net or a sum contribution. It's a sum contribution of 1 potassium efflux, which acts to hyperpolarize the cell, as well as another contribution of sodium and calcium influx. These act to depolarize the cell. Then the end result, or the net result, is that this cell will get depolarized and it will remain in the state of negative 40 millivolts, which is a constant value, and it's known as dark current. So remember, these channels are kept open by cyclic GMP, which increases in concentration and binds there. This is due to the action of guanyl cyclase, as we have mentioned there. And from the beginning, I said when these cells are depolarized, they release neurotransmitters. Now that this cell is depolarized, if you take a look at this synaptic terminal here, like the way I've zoomed there. Remember, when neurons that have got neurotransmitters get stimulated by and action potential has been generated in those neurons, what happens is that at the synaptic terminal there are voltage gated calcium channels there. So this is not different from photoreceptors. Photoreceptors also at the synaptic terminal have got vesicles there that have got neurotransmitters, and they also have got voltage gated calcium channels. So since they are in a depolarized state, like we have mentioned here, this receptor potential that is generated, this dark current that is generated here, causes the opening of these voltage gated calcium channels, and therefore calcium can influx into the cell there, like shown there. Calcium can influx there, and calcium influx is there as well. The influx of this calcium causes the release of this neurotransmitter, which is glutamate in this case. Like I mentioned here, I said rod synapse with bipolar cells. So this is a synapse. These are bipolar cells. This one is a rod synaptic terminal. So there's a release of glutamate from the rods into the synaptic cleft between the bipolar cells and the rods. So let's just run through what we have talked about before we proceed. So I'm saying we're talking about what happens during the dark when there's no light stimuli, and I've said guanyl cyclase in the membranous disc there gets to convert GTP into cyclic guanosine monophosphate, which increases during the dark. And when this increases, what it does is that it binds onto these channels here, the cyclic guanosine monophosphate gated channels, binds there and keeps them open. And I've said these receptors, the rods in the dark are depolarized. They have got a membrane of about negative 40 millivolts. The depolarization is due to the following. So the first thing I've talked about is that they are in the inner segment. You have the sodium potassium pump ATPase there, or sodium potassium pump. It leaks out 3 sodium ions and leaks into potassium ions. This is just to maintain the concentration of these ions inside the cell. Then this leaked sodium goes back into the cell through the outer segment there, and it goes through these cyclic guanosine monophosphate gated channels, which are kept open by binding of cyclic guanosine monophosphate there. The other ion that leaks in is calcium ion. This acts to depolarize the cell. Now at the same time, potassium is leaking out in the inner segment through these non-gated potassium channels. This function acts to oppose this depolarization by hyperpolarizing the cell. And I'm saying, what I've said is that the net contribution of this hyperpolarization by the potassium which leaks out through the inner segment through this channel, and the depolarization that happens through the influx of calcium and potassium, the net contribution results in the depolarization of these cells to the membrane of about negative 40 millivolts. And due to that depolarization, there's a synaptic cleft, synapse here between the rods and the bipolar cells. And I've said due to this depolarization, or this receptor potential that is generated by this whole scenario here, it gets to open these voltage-gated calcium channels that are on the synaptic terminal. When these open, the calcium ions influx, and when these calcium ions influx, they cause the release of the neurotransmitter glutamate here between the rods and the bipolar cells. Now, as we all know, the glutamate is actually, which is generally an excitatory neurotransmitter. But in this case, glutamate is inhibitory on the bipolar cells. So when glutamate has been released there, this glutamate released here is inhibitory on the bipolar cells. It's inhibitory on the bipolar cells. So it gets to inhibit the release of glutamate. It gets to inhibit these bipolar cells and thereby inhibit release of the neurotransmitter glutamate here between the bipolar cells and the ganglionic cells. So due to the fact that there is no release, or there is less release, causes less release of glutamate on the synapse between bipolar cells and ganglionic cells. So what I was just trying to say, that less glutamate or no glutamate is released on this synapse between the bipolar cells and the ganglionic cells. Due to that, these ganglionic cells are not stimulated, and therefore an action potential is not generated. So the ganglionic cells are not stimulated since there's less or no release of glutamate onto them, onto their dendrites, or the processes are not stimulated. Therefore, action potentials are not generated in the ganglionic cells. Remember I said rods and bipolar cells, you have receptor potentials in here. But with the ganglionic cells, that's why action potentials are not generated. But now, due to the fact that this glutamate released by the rods is inhibiting these bipolar cells, less or no glutamate is released on these ganglionic cells. Since there's less or no glutamate is released on these ganglionic cells, less or no action potentials are generated in the ganglionic cells, and therefore perception of vision is not going to occur. So this is what happens in the dark.